What if I told you that um, every time you recall a memory like your first kiss, a childhood trauma, or even just, how to ride a bicycle, your brain literally demolishes the physical microscopic structure of that memory?
Yeah, tears it apart right down to the proteins, and then in real time, it has to frantically rebuild it from scratch before you forget it forever.
Right. And if anything interrupts that reconstruction process, the memory doesn't just fade, it's permanently erased.
It really fundamentally changes how we view our own minds. I mean, we tend to think of memory as like a video recording stored on a hard drive somewhere in our
Yeah,
heads.
like you pull the file up, you watch it, you put it back.
Exactly. But the biological reality is that memory is this living, breathing, physically dynamic architecture that is just constantly being destroyed and rebuilt.
And that structural reality is exactly what we are exploring today. We are taking a deep dive into Chapter 53 of the 6th edition of Principles of Neuroscience.
Such a massive foundational text.
It really is. And our mission today is to sit down, kind of as if we were in an advanced graduate-level neuroscience seminar, and just rigorously unpack the cellular mechanisms of implicit memory storage.
We want to understand, literally molecule by molecule, how your unique daily experiences physically alter the neural circuits of your brain.
Right, to change your behavior, and ultimately how those physical changes form the actual biological basis of your individuality.
To start that journey, though, we have to recognize a really fundamental division in how the brain handles information.
Okay, lay it out for us.
We basically must separate the memories you can consciously declare from the memories that your body just simply executes. We categorize these as explicit memory and implicit memory.
So explicit memory is the conscious kind, right? Like people, places, objects, facts.
Right. If I ask you to recall the name of your first grade teacher or, what you ate for dinner last night, you're actively searching for and retrieving an explicit memory.
And structurally, we know that explicit memory relies heavily on a specific area of the brain, the hippocampus, and the medial temporal lobe, if I'm not mistaken.
You're spot on. The hippocampus acts as the initial cataloging system for these conscious memories before they get, distributed out to the broader neocortex for long-term storage.
But implicit memory operates in an entirely different universe, doesn't it?
Completely different universe. Implicit memory is totally unconscious and automatic. It encompasses your conditioned responses, your deeply ingrained habits, and your perceptual and motor skills.
So, like when you type on a keyboard without looking at your fingers.
Exactly. Or when your heart suddenly races because you hear a siren that sounds exactly like the one from a car accident you were in years ago. That is you utilizing implicit memory.
Because it's a different system, I assume it relies on a completely different set of brain structures.
It requires a highly diverse family of neural circuits. The neocortex is involved in perceptual priming. The striatum is the main hub for executing your complex motor skills.
Okay, and the amygdala.
The amygdala, which we will spend a lot of time on today, is the epicenter for Pavlovian threat conditioning.
Right. And then there's the cerebellum for fine-tuning learned motor movements.
Yeah, and for the absolute simplest forms of learning, the memory doesn't even require a specialized brain structure at all. The memory is stored directly in the reflex pathways themselves.
Which is wild to think about, and to truly grasp how isolated these two systems are from each other. Our source material highlights two incredibly striking clinical cases.
Oh, these cases are just fascinating.
They really are. The first is Clive Waring. He was a highly accomplished English musician and conductor, and in 1985, he contracted a severe case of herpes encephalitis.
Which is a viral infection that attacks the brain.
Right. The virus attacked his brain and completely devastated his hippocampus and his temporal cortex. The aftermath was this total, catastrophic loss of his short-term explicit memory.
It's one of the most profound cases of amnesia ever recorded. His conscious timeline was effectively erased every few moments.
Yeah, he could not remember a conversation he had literally 2 minutes prior.
Yet, despite having his conscious memory completely wiped, his implicit memory system remained remarkably intact.
Which is the crazy part. If someone guided him to a piano bench, he could sit down, read complex sheet music, play beautifully, and even conduct a chorale.
With the exact same mastery he had before the illness. His procedural memory, his deeply ingrained musical skills was perfectly preserved in the striatum and motor cortex.
But the tragic paradox is that the moment the music stopped and the performance ended, he couldn't explicitly remember having just played the piano.
The knowing how survived, while the knowing what was completely destroyed.
Wow. And the second case provides a similar clinical proof from a totally different angle, right? William de Kooning.
Right, the famous abstract expressionist painter. Later in life, he developed severe Alzheimer's disease.
Which is a neurodegenerative condition that viciously attacks the hippocampal and cortical networks responsible for explicit memory.
Yes. He lost his ability to remember people, he forgot places he had lived, and he really lost his grasp on recent events.
Yet, as the Alzheimer's progressed and his conscious mind deteriorated, he continued to paint.
And he didn't just casually paint. He produced masterful, highly valued works of art.
Because the creative, skill-based motor programs and the implicit aesthetic rules he had spent a lifetime developing were entirely insulated from the decay of his conscious memory.
Exactly. They were running on different hardware.
So trying to visualize this, is explicit memory like saving a text document to a computer's hard drive? Like it's a file that can be corrupted or deleted if the physical drive takes a hit.
Okay, yeah.
While implicit memory is more like changing the physical wiring on the motherboard itself. So even if the hard drive with the text documents is wiped, the machine's motherboard is still physically wired to know how to process graphics or run the cooling fans.
It's a really helpful analogy for understanding the separation of the systems, but we actually need to refine the biological premise just a bit.
Okay, correct me.
The fundamental truth of neuroscience is that all learning, whether it's memorizing a poem or learning to hit a tennis ball, ultimately alters the effectiveness of synaptic transmission.
So all of it involves physical changes to the motherboard, to use my phrasing.
Yes. Every memory is a structural change, so explicit memory changes the motherboard too.
Got it. So what's the actual difference then?
The difference lies in the location and the function of those changes. With explicit memory, the physical changes occur in massive, highly complex associational networks in the hippocampus and cortex that represent abstract concepts.
Right, the facts and relationships.
But with implicit memory, particularly in the simpler forms we are going to break down today, the learning physically alters the very reflex pathways that directly execute the behavior itself.
Ah, I see.
The memory isn't a file that is retrieved by the circuit. The memory is the physical shape and strength of the circuit.
The synapse itself holds the memory. That makes so much sense. To understand exactly how a synapse changes shape based on experience, we have to look at the absolute simplest forms of learning.
Right. We need to look at non-associative learning, specifically habituation and sensitization.
Let's start with habituation.
Habituation is the most fundamental form of implicit learning. It is simply learning to ignore a novel stimulus because it turns out to be harmless.
Like learning to tune out the hum of a refrigerator.
Exactly. Or imagine a snail crawling on the sidewalk. If you gently tap its shell, it immediately retracts into the shell. That's an innate defensive reflex.
Right.
But if you sit there and tap the shell softly every 10 seconds, the snail's nervous system realizes that the tap is not followed by a predator trying to eat it.
The stimulus is irrelevant.
So the snail stops retracting. It habituates.
And it's crucial to note here that this isn't muscle fatigue, right? The snail isn't just too tired to pull back.
No, not at all. The nervous system is actively suppressing the response. The physiological foundation for this was actually documented over a century ago by Charles Sherrington.
Oh, the guy who studied cats.
Yes. He was studying the posture and locomotion reflexes in cats. Sherrington would electrically stimulate certain sensory pathways that caused a motor reflex in the cat's leg.
And he noticed that if he stimulated the same pathway repeatedly, the intensity of the muscle contraction steadily decreased.
Right. He theorized that this behavioral habituation was caused by a functional decrease in the effectiveness of the synapse connecting the sensory neuron to the motor neuron.
He literally guessed that the connection was getting weaker.
And modern cellular biology proved him right. We now call this presynaptic depression. The sensory neuron simply stops releasing as much neurotransmitter.
Okay, so that's habituation. Sensitization, on the other hand, is the biological opposite, right?
It is. Sensitization occurs when an animal experiences a severe noxious stimulus, and as a result, it develops a globally heightened response to all stimuli, even completely harmless ones.
The classic example is walking through a dangerous neighborhood at night. If you suddenly hear a gunshot ring out nearby, your sympathetic nervous system flares up.
You are highly sensitized.
Yeah, and if a stray cat brushes against your leg 10 seconds later, you don't just notice it. You jump completely out of your skin.
Your defensive reflexes are hyper-amplified by the prior noxious event.
So, to understand the exact molecular play-by-play of how habituation and sensitization physically happen at a synapse, neuroscience owes an enormous debt to a very specific creature.
The marine invertebrate Aplysia californica, commonly known as the sea slug.
Now, I have to ask, if we are trying to understand human memory, studying a giant squishy sea slug might seem like a totally bizarre detour. Why not study a mouse or a primate from the start?
It's a fair question. It really comes down to the deep evolutionary conservation of survival mechanisms. Anticipatory defensive responses, the ability to learn what in your environment is dangerous and what is safe, are an absolute requirement for the survival of almost any moving organism.
Because if you can't learn that, you get eaten.
Exactly. Because this need is so fundamental, nature evolved a highly effective molecular mechanism for it millions of years ago, and then rigidly conserved that mechanism across the animal kingdom.
So the chemical cascade in a slug is shockingly similar to the cascade in a human.
It is. But the slug is just infinitely easier to map. The human brain has roughly 86 billion neurons forming trillions of overlapping convoluted synaptic connections.
Which is a nightmare to try and trace.
Right. The Aplysia has only about 20,000 neurons in its entire central nervous system. Furthermore, these neurons are unusually large. Some can actually be seen with the naked eye.
Wow, really?
Yeah. And they are consistently located in the exact same anatomical position from one slug to the next. This allowed researchers, most notably Eric Kandel and his colleagues, to map the exact specific neural circuit of a behavior, cell by individual cell.
Okay, so the specific behavior they focused on is the gill-withdrawal reflex. Let's describe the anatomy. It's pretty simple.
Right.
The slug has a respiratory organ called a gill, which sits exposed on its back. Next to the gill is a small fleshy spout called a siphon, which it uses to expel seawater.
And if you physically touch the siphon, the slug reflexively pulls its fragile gill inward to protect it from potential harm.
So how does that simple reflex map out neurologically?
It is a remarkably direct neural circuit. Sensory neurons innervate the skin of the siphon. When the skin is touched, these sensory neurons fire electrical signals, called action potentials, down their axons.
And where do they go?
These sensory axons make direct monosynaptic connections onto the motor neurons that control the muscle fibers of the gill.
Let's slow down and look at that exact junction. When the action potential reaches the end of the sensory neuron, what physically happens to cross the gap to the motor neuron?
This is where the chemistry happens. As the electrical action potential arrives at the presynaptic terminal of the sensory neuron, the shift in voltage causes specialized channels in the cell membrane to open.
These are the voltage-gated calcium channels, right?
Yes. Calcium ions from the outside fluid rush into the terminal. This sudden influx of calcium is the critical trigger. It causes tiny molecular sacs called vesicles, which are packed full of a neurotransmitter, specifically glutamate, to fuse with the cell membrane.
And then they just dump their glutamate into the synaptic cleft.
Exactly. The glutamate floats across the gap and hits the motor neuron.
And what happens on the receiving end?
The glutamate binds to receptors on the postsynaptic membrane of the motor neuron. This binding opens ion channels on the motor neuron, allowing sodium to rush in.
Which creates a charge.
Right. It creates a slight positive electrical charge called an excitatory postsynaptic potential, or EPSP. If the sensory neuron dumps enough glutamate, the EPSP in the motor neuron reaches a critical threshold. The motor neuron fires its own action potential, the gill muscle contracts, and the gill withdraws.
That makes perfect sense. That is the baseline reflex. Okay, so that's the resting state. Now, let's apply Sherrington's concept of habituation to the slug.
Okay.
If the researcher repeatedly touches the siphon with a brush every minute, the slug learns the brush is harmless and stops withdrawing the gill. What is actually happening to the calcium and the glutamate?
As the sensory neuron fires action potentials repeatedly over a short period, a temporary molecular exhaustion occurs at the terminal. Specifically, fewer and fewer voltage-gated calcium channels open in response to the action potential.
Do we know why they get exhausted?
We still don't know the precise mechanism of why the calcium channels become inactivated, actually, but the result is crystal clear. Less calcium flows into the presynaptic terminal.
And since calcium is the trigger that pushes the vesicles to the membrane, less calcium means less glutamate is dumped into the gap.
Exactly. The sensory neuron still fires fully, but its output is muted. Because less glutamate hits the motor neuron, the resulting EPSPs in the motor neuron get smaller and smaller.
Until they just fizzle out.
Pretty much. Eventually, the EPSP is so weak that it fails to push the motor neuron to the firing threshold. The motor neuron stays silent, and the gill does not move.
So the memory of habituation is stored physically as a temporary decrease in synaptic effectiveness, presynaptic depression.
Yes. It's a very elegant energy-saving measure. The system just dials down the volume knob on the neurotransmitter.
So what about sensitization? If you give the slug a noxious shock to its tail, it suddenly becomes terrified of everything.
Yeah.
If you gently touch the siphon now, it violently yanks its gill inside.
Right.
How does a shock to the tail affect a synapse located way over between the siphon and the gill?
It requires an intermediary. Sensitization introduces a third party into the circuit. When you shock the slug's tail, sensory neurons in the tail fire aggressively.
Okay.
These tail neurons don't just connect to tail muscles, they also project into the central neural circuitry and activate a special class of cells called facilitatory interneurons.
They act like a localized alarm system.
Precisely. These facilitatory interneurons cast a wide net. They send their axons to form synapses directly onto the presynaptic terminals of the siphon sensory neuron.
Wait, a synapse on a synapse?
Yes. This is a unique anatomical setup called an axoaxonic synapse. It's a synapse landing directly on the broadcasting end of another synapse.
So the interneuron is physically in a position to dictate how the sensory neuron behaves.
When the tail is shocked, these facilitatory interneurons fire and release a neuromodulator onto the terminal of the sensory neuron. The primary neuromodulator in this pathway is serotonin.
Serotonin, okay, that's a big one.
Yes. The serotonin binds to metabotropic receptors embedded in the membrane of the sensory neuron's presynaptic terminal. This binding sets off a really complex biochemical cascade inside the sensory neuron.
Walk us through that cascade. What does the serotonin actually change inside the terminal to make it so hyperreactive?
The serotonin cascade ultimately targets potassium channels. Normally, after an action potential peaks, potassium channels open, allowing potassium ions to rush out of the cell.
And why does the potassium rush out?
This rapid exit of positive charge is what brings the cell's voltage back down to its restive state, ending the action potential. It's the repolarization phase.
So potassium is essentially the brake pedal for the action potential.
Exactly. But the serotonin cascade causes those potassium channels to become sluggish. They close slowly or fail to open fully.
Uh-oh. So the brake pedal is broken.
Right. Because the brake is broken, the action potential doesn't end quickly. It is significantly prolonged. The terminal stays positively charged for a much longer duration.
And if the terminal stays positively charged longer, that means the voltage-gated calcium channels stay open longer, right?
That is the crucial link. The prolonged action potential means the calcium channels stay wide open. A massive flood of calcium rushes into the terminal.
And as we established earlier, calcium triggers the glutamate vesicles.
Yes. With all that extra calcium, a massive, excessive amount of glutamate is dumped into the synaptic cleft.
So let me trace this back. The next time you give the siphon a completely harmless, gentle tap, the sensory neuron fires. But because it's sensitized by the serotonin, it unleashes an absolute torrent of glutamate.
Yep. The motor neuron receives a massive EPSP, fires violently, and the gill is yanked back with extreme force.
That is presynaptic facilitation. So the memory of the tail shock is temporarily stored as a biochemical alteration of the potassium channels in a completely different sensory pathway.
You've got it. Habituation is the sensory neuron getting lazy with its calcium, and sensitization is serotonin forcing the sensory neuron to leave its calcium gates wide open.
That fundamentally demystifies non-associative learning. It's just incredible. But memory in humans is rarely that simple, right? We form associations. We learn that event A predicts event B.
Right, like Pavlov's dog learning that a ringing bell predicts meat powder.
Exactly, classical conditioning.
Now, a brief note on terminology here based on our source text. In the past, when an animal learned to associate a neutral stimulus with a painful one, the field broadly referred to it as fear conditioning.
Right.
However, rigorous modern neuroscience has shifted to calling it threat conditioning.
Because we can't definitively know if a rat or a sea slug actually experiences the subjective, conscious emotion of fear the way a human does, right?
Exactly. Attributing complex human emotional states to simple organisms without proof is a pitfall. What we can observe and measure objectively is their physiological and behavioral defense response to a learned threat. So, threat conditioning is the accurate, objective term.
Makes sense. So how do we threat condition our sea slug?
You apply a classical conditioning protocol. You introduce a conditioned stimulus, a very light, non-threatening tap to the siphon, and you pair it with an unconditioned stimulus, a strong shock to the tail.
Okay.
But the timing must be perfectly orchestrated. The siphon tap must precede the tail shock by roughly half a second.
So the brain has to perceive the tap as the warning siren for the shock.
Exactly. If you repeat this pairing a few times, the Aplysia learns the association. The tap predicts the shock. And structurally, this associative learning results in a facilitation of the gill-withdrawal reflex that is significantly more powerful and lasts much longer than the simple sensitization we just discussed.
But wait, hang on. If sensitization already floods the terminal with serotonin, what does pairing the tap and the shock actually add to the equation? How does the cell know it's experiencing an association, rather than just a generalized shock?
That is the million-dollar question.
Yeah.
It utilizes an incredibly sophisticated molecular mechanism called coincidence detection. Let's look inside the presynaptic sensory neuron terminal during the exact moment of conditioning.
Okay, painting the picture.
First, you tap the siphon. The sensory neuron fires an action potential, which means a small amount of calcium enters the terminal. Half a second later, the tail shock occurs, causing the inner neurons to dump serotonin onto that very same terminal.
So both events are physically overlapping inside the terminal at the same time?
Yes. The serotonin binds to its receptor and activates an enzyme inside the terminal called adenyl cyclase.
Adenyl cyclase.
Yes. It's the enzyme responsible for creating the intracellular signals that lead to facilitation. And here is the coincidence detection. Adenyl cyclase is designed in such a way that it is stimulated by serotonin, but its activity is massively amplified if it is simultaneously exposed to calcium.
Oh, wow. So if the shock, meaning the serotonin, happens while the terminal is completely quiet, the enzyme works at a baseline level.
Right.
But if the shock happens immediately after a tap, when the terminal is freshly flooded with calcium, the enzyme just goes into absolute overdrive.
Exactly. The convergence of calcium from the sensory firing and serotonin from the shock causes adenyl cyclase to produce a massive surge of intracellular signaling molecules. The cell has molecularly detected the coincidence of the two stimuli. The presynaptic facilitation is hugely amplified.
That perfectly explains how the sensory neuron pushes harder. But does the motor neuron on the receiving end play a role in this association, or is it all presynaptic?
It absolutely does. There is a vital postsynaptic component, too. During classical conditioning, the tap and the shock occur close together. The tap causes the sensory neuron to release glutamate. The shock causes the motor neuron to become generally excited and partially depolarized.
Yeah.
If the sensory neuron releases a large amount of glutamate precisely when the motor neuron is already highly depolarized, it causes a specific voltage-dependent channel on the motor neuron to open, allowing a massive influx of calcium into the motor neuron itself.
So the presynaptic cell fills with calcium because of the coincidence, and the postsynaptic cell fills with calcium because of the coincidence.
Yes. This postsynaptic calcium triggers a backward-traveling chemical signal that reaches back across the cleft to further enhance the sensory neuron's neurotransmitter release. The synapse is literally strengthened from both sides simultaneously.
That's incredible.
And what is really fascinating is that this postsynaptic calcium mechanism in the slug is actually the evolutionary precursor to long-term potentiation, which is the mechanism mammals use for complex learning, and we will detail that later.
So that is how the association is structurally encoded in the short term. But here is the critical pivot point of this whole discussion.
Okay.
Everything we've discussed so far, the calcium influx, the sluggish potassium channels, the ester glutamate, these are all temporary chemical flutters. The proteins eventually reset. The effect lasts for minutes, maybe an hour or two.
Correct.
But memories can last a lifetime. So how does practice make perfect? How do we translate a temporary chemical spike into a permanent structural change in the architecture of the brain?
To understand the shift from short-term to long-term memory, we must explore the cAMP-PKA-CREB pathway. This biochemical cascade is the physical machinery of long-term consolidation.
The cAMP-PKA-CREB pathway.
Yes. In the Aplysia, a single tail shock produces sensitization that fades within an hour. But if you expose the slug to spaced repetition, say 5 separate tail shocks spaced out over a few hours, the sensitization lasts for days.
Okay.
And if you provide further spaced training over several days, the memory persists for weeks or even months.
Every student cramming for an exam knows this, right? Reading a chapter 5 times in 1 hour is useless compared to reading it once a day for 5 days. The brain demands spaced repetition.
It truly does.
But what is the cellular difference? What happens when serotonin hits the receptor 5 times spaced out instead of just once?
It is a matter of accumulation crossing a critical threshold, which fundamentally changes where the signaling occurs within the cell. Let's trace the full cascade. When serotonin binds to the receptor on the sensory neuron, it activates a G protein inside the membrane.
Okay.
This G protein activates adenyl cyclase, the enzyme we just discussed. Adenyl cyclase grabs molecules of ATP, the standard energy currency of the cell, and converts them into cyclic AMP, or cAMP.
cAMP, that's the famous second messenger, right? It carries the news of the serotonin deeper into the cell.
Precisely. cAMP then binds to and activates an enzyme called protein kinase A, or PKA. Now, PKA is complex. It's composed of two different subunits stuck together, a regulatory subunit and a catalytic subunit.
Okay, two parts.
When they are joined, the enzyme is turned off. But when cAMP binds to the regulatory subunit, it forces it to change shape and release the catalytic subunit.
So the catalytic subunit is unleashed into the cell.
Yes. The free catalytic subunit is now active. A kinase's job is to phosphorylate other proteins, meaning it attaches a phosphate group to them, which changes their biological function.
Got it.
During short-term memory, generated by a single shock, a small amount of cAMP frees a small number of catalytic subunits, and they stay right there in the local presynaptic terminal, phosphorylating the potassium channels to close them.
But with spaced repetition, the system is flooded, I imagine.
Flooded. With spaced repetition, the relentless waves of serotonin cause cAMP levels to spike dramatically and stay elevated for a prolonged period. This frees a massive army of PKA catalytic subunits.
And where do they go?
When the concentration gets high enough, they don't just stay in the terminal, they translocate. They physically travel all the way up the axon, traversing the entire length of the cell, and they enter the nucleus.
Oh, wow. They cross the nuclear envelope and access the DNA itself.
Yes, because the conversion of a short-term memory to a long-term structural memory requires the construction of new physical synapses. To build new structures, you need new proteins. And to get new proteins, you must activate gene expression.
Right.
Once inside the nucleus, the PKA catalytic subunit finds a specific transcription factor called CREB1, which stands for cAMP response element binding protein 1. PKA phosphorylates C1, turning it on.
So C1 is like the master switch for memory consolidation.
Exactly. Activated CREB1 binds to a specific sequence of DNA called the CRE element. It recruits a coactivator called CBP, which acts like a beacon for RNA polymerase II. RNA polymerase II lands on the DNA and begins transcribing the specific genes that code for the structural proteins needed to grow new synaptic connections.
Okay, so CR1 essentially hits the gas pedal on protein manufacturing.
It does, but biological systems are extremely cautious. Growing new synapses requires a massive amount of metabolic energy. The cell cannot afford to consolidate every passing trivial event into long-term memory.
That makes sense. We'd run out of energy or space.
Right. Therefore, the system has a built-in parking brake. That brake is another transcription factor called CREB2. CREB2 binds to the same DNA regions, but instead of activating transcription, it represses it. It physically blocks the process.
Oh, interesting. So if the cell wants to commit something to long-term memory, hitting the gas isn't enough. It has to simultaneously remove the parking brake.
Exactly. And the repeated training achieves this. At the same time that PKA is entering the nucleus to activate CREB1, the intense, prolonged serotonin signaling indirectly activates a completely different kinase pathway called MEPK mitogen-activated protein kinase.
And what does MEPK do?
MEK also enters the nucleus, and its target is CREB2. When MAPK phosphorylates CREB2, it suppresses the repressor. It forces CREB2 to detach from the DNA.
So PKA hits the gas by activating CREB1, and MAPK simultaneously releases the parking brake by removing CREB2.
Yes, and this dynamic interplay between an activator and a repressor provides a really profound insight into human psychology, if you think about it.
How so?
The exact balance of these two factors acts as a threshold. It explains why our ability to form long-term memories fluctuates so wildly in everyday life. Think about it. The ease with which an experience crosses the threshold into long-term memory is heavily dictated by your attention, your emotional state, and the context of the event.
Oh, that makes so much sense.
A traumatic experience or a moment of extreme joy floods the brain with neuromodulators that effortlessly remove the CRB2 brake and floor the CRB01 gas pedal, consolidating the memory instantly.
While reading a boring textbook requires you to manually force the cascade through tedious spaced repetition just to get enough PKA to overcome the baseline repression.
Precisely.
Now, the source material also introduces an epigenetic component here, noncoding RNAs. How do they fit into this switch?
This is one of the most exciting recent discoveries. For decades, the dogma of biology was that DNA's primary job was to code for messenger RNA, which then coded for proteins. But we now know the genome produces vast amounts of small RNA molecules that do not code for proteins at all.
What do they do instead?
They are regulatory. They dictate whether other messenger RNAs are allowed to be translated. Serotonin signaling dynamically orchestrates these non-coding RNAs to ensure the memory takes hold.
How does the serotonin interact with these RNAs exactly?
It acts in two distinct phases. Immediately after serotonin hits the cell, there is a rapid downregulation of a specific microRNA called miRNA-124. In a resting cell, miRNA-124 acts as a suppressor. It prevents the translation of CREB1 messenger RNA into the actual CREB1 protein.
So by rapidly decreasing miRNA-124, serotonin removes a roadblock.
Yes, allowing a sudden burst of CREB1 protein to be manufactured, facilitating the initial wave of gene transcription.
It clears the supply lines. And what's the second phase?
After a delay, the sustained serotonin signaling actually increases the production of a different non-coding RNA called purNAF. purNAF forms a complex with other proteins, and this complex physically enters the nucleus and targets the promoter region of the CREB2 gene, the parking brake gene.
And what does it do to the brake?
It chemically modifies the DNA through a process called methylation. DNA methylation permanently silences the gene. It physically shuts down the factory that produces the CRDB2 repressor.
Wow, so it's not just a temporary shift in the balance. The cell is epigenetically rewriting its own manufacturing code to permanently silence the repressor. It's locking the cell into a pro-memory, pro-growth state.
Exactly.
Okay, but all of this nuclear activity raises a massive logistical problem in my mind.
The specificity problem.
Exactly. Let's say I'm learning a highly specific fine motor skill. Only a handful of synapses need to be strengthened. The nucleus of the neuron is sitting in the cell body, acting as a central factory. It gets the CRB1 signal, and it pumps out thousands of new mRNAs and proteins.
Right.
But a single mammalian neuron might have 10,000 different synaptic connections branching off its axon, connecting to thousands of different target cells. When the factory ships these new memory proteins out into the axon, how do the proteins know which of the 10,000 synapses they're supposed to rebuild? Why doesn't the whole neuron just grow everywhere indiscriminately?
It is perhaps the most fundamental question in the cellular biology of memory. How is long-term memory synapse-specific? The biological answer is a phenomenon known as synaptic capture.
Synaptic capture.
Yes. The definitive proof of this came from a beautifully designed experiment by Kelsey Martin and her colleagues. They created a highly specialized in vitro system. They extracted a single sensory neuron from an Aplysia and placed it in a culture dish.
Okay.
They allowed its axon to grow, but they manipulated it so that the axon bifurcated it, split into two distinct branches, forming an upside-down Y shape.
Okay, so one cell body, one main axon splitting into two separate output branches.
Right, and they arranged it so that branch A formed synaptic connections with one motor neuron, and branch B formed connections with a completely different motor neuron.
Got it.
They then took a microscopic pipette and carefully applied 5 pulses of serotonin exclusively to the synapses at branch A. The synapses at branch B were left completely alone.
Okay, based on the cascade we just discussed, the 5 pulses at branch A should trigger the heavy machinery. The cAMP should spike locally. PK should travel all the way back up the axon from branch A to the nucleus, activate CREB1, and start transcription.
And that is exactly what happened. The nucleus began manufacturing new proteins and mRNAs essential for synaptic growth. And this is where the critical observation occurred. As these new materials were shipped out of the cell body and traveled down the main axon, they didn't just go to branch A.
Where did they go?
They flowed indiscriminately down both branches. They arrived at the synapses of branch A, and they arrived at the quiet synapses of branch B.
But wait, if the building materials arrived at both sites, why didn't both synapses grow?
Because merely having the building materials isn't enough. Long-term facilitation, the actual structural growth of the synapse, only occurred at branch A. The synapses at branch B remained unchanged despite being bathed in the exact same new proteins.
What permitted branch A to build?
The initial localized application of serotonin at branch A did 2 things simultaneously. It sent the signal to the nucleus, yes, but it also created a local biochemical tag at the specific synapses of branch A.
Ah, a tag.
Yes. This tag altered the local molecular environment of those specific synapses. Only synapses that have been tagged by recent, highly specific local activity possess the biochemical clearance to capture the newly arrived proteins and utilize them to build new structure.
Oh, that is brilliant.
Yeah.
So the nucleus blindly ships the lumber and the bricks to every construction site in the city. But only the specific foremen who experience the local serotonin spike have the architectural blueprints required to actually use the bricks to build a wall.
That's a fantastic way to put it.
And all the other sites just let the bricks sit there until they degrade.
It is an incredibly efficient system, and Martin proved the mechanism with a second, brilliant variation of the experiment.
What did she do?
She again applied the 5 pulses of serotonin to branch A, initiating the massive nuclear production of proteins. But this time, while the proteins were traveling down the axon, she applied a single pulse of serotonin to branch B.
Now, a single pulse shouldn't cause long-term memory. It only causes short-term facilitation lasting minutes because it's not enough to send pKa to the nucleus.
Exactly. On its own, a single pulse does not activate gene expression. But in this scenario, the proteins had already been manufactured due to the activity at branch A.
Ah.
The single pulse at branch B was sufficient to create the local biochemical tag at those synapses. Because the tag was now present and the proteins happened to be floating by, branch B captured the proteins and successfully underwent long-term structural facilitation.
That is mind-bending. The memory was initiated by one pathway, but another pathway was able to hijack the building materials just by being slightly active at the exact right moment.
Yes, it perfectly explains the associative nature of memory on a massive scale.
But there is another logistical problem that we have to address. We are talking about memories that last for decades. But memory relies on proteins. Proteins are biological molecules subject to entropy. They degrade.
They absolutely do.
The structural proteins that make up a synapse have a half-life of perhaps a few days or weeks. How can the physical structure of memory remain stable for a lifetime if the physical building blocks it is made of are constantly disintegrating?
This conundrum led to one of the most startling and revolutionary discoveries in molecular neuroscience. The mechanism that maintains the memory over the long term involves a specific regulatory protein called CPEB.
CPEB, which stands for
Cytoplasmic polyadenylation element binding protein. CPEB acts as a local manager of protein synthesis right at the synapse. But what makes CPEB truly extraordinary and what initially shocked the scientific community is that its active form behaves exactly like a prion.
Wait, hold on. You say prion, and anyone who knows biology immediately thinks of mad cow disease or Creutzfeldt-Jakob disease. Prions are terrifyingly destructive, misfolded proteins that clump together and literally turn the brain into a sponge. Are you saying a disease mechanism is responsible for memory?
I know, it sounds alarming. It is the exact same structural mechanism, but with a profoundly different biological outcome. Pathogenic prions, like those responsible for mad cow disease, are toxic because their aggregated clumped form is entirely unregulated and it destroys cellular function, eventually killing the neuron.
Right.
But the CPEB found in the Aplysia and its homologs in mammals is a functional, non-pathogenic prion. It is a feature, not a bug.
How does a functional prion work without killing the cell?
In a naive resting synapse, CPEB exists in a soluble, inactive state. It just floats around doing nothing. However, when the synapse is subjected to intense serotonin signaling during the space learning trials, it triggers the local synthesis of more CPEB protein.
Okay, so it builds up.
As the concentration of CPEB at that specific synapse rises, it hits a critical threshold. At this threshold, the CPEB proteins undergo a dramatic conformational change. They physically alter their 3D shape and begin to bind to each other, forming a large, insoluble aggregate.
It clumps together, just like the disease prions.
Yes, but here, the aggregated clump is the highly active form of the protein. And because of its fundamental prion nature, the aggregate is self-perpetuating.
Meaning it sustains itself.
Exactly. Once the clump forms, it acts as a permanent structural template. As old CPEB molecules degrade, the aggregate magnetically attracts any newly synthesized soluble CPEB molecules that happen to float by, and forces them to adopt the misfolded shape and join the clump.
It's like a specialized Lego block. Once you snap the first few pieces together to form a pillar, the magnetic shape of the pillar forces any other passing Lego blocks to instantly snap into place, ensuring the pillar never degrades. It constantly refreshes its own structure.
That's a perfect analogy.
But what does this permanent CPEB pillar actually do for the memory?
The active CPEB aggregate has a very specific job. It binds to dormant messenger RNAs that are stored in the presynaptic terminal. These mRNAs are just sitting there, unable to be translated.
Okay.
The CPEB aggregate recruits an enzyme that attaches a long chain of adenine nucleotides, a poly-A tail, to these mRNAs. In cellular biology, adding a long poly-A is the equivalent of waving a massive green flag for the ribosomes. It signals the ribosomes to immediately attach to the mRNA and begin translating it into protein.
And what proteins are being translated right there at the synapse?
Specifically, the structural proteins required to maintain the size and strength of the synapse, like actin and tubulin. Because the self-perpetuating cPEB prion is a permanent fixture at the synapse, it creates a constant, localized, unending factory that churns out structural beams and girders year after year.
Wow.
The synapse maintains its expanded strength and shape completely independently of the nucleus because the local prion manager ensures the structural integrity is constantly renewed.
That completely reframes the permanence of memory. A localized, self-sustaining prion engine. But as permanent as that sounds, we also know that memory is fallible. It's malleable.
Right.
Which brings us back to the idea I mentioned at the very start of this deep dive, the idea that remembering something destroys it.
Yes. For decades, the dominant theory of memory consolidation was that once the genetic cascade finished and the structural changes were locked in, the memory was fixed. It was stable. Remembering it was just accessing a read-only file.
Right, like pulling a book off a shelf.
But the neuroscientist Karim Nader upended the entire field with the discovery of memory reconsolidation. Nader demonstrated that the very act of retrieving a memory makes the physical trace of that memory temporarily fragile and unstable all over again.
Why would the brain evolve to do that? Making a vital survival memory fragile every time you use it seems incredibly dangerous.
It's a trade-off for flexibility. If an animal remembers the location of a water source, but the environment changes and a predator moves into that area, the animal needs to update the memory with new context.
Oh, I see.
If the memory were rigidly locked in concrete, it couldn't adapt. Nader showed that retrieving a memory requires a fresh, rapid wave of protein synthesis to restabilize the synapse, a process he termed reconsolidation.
And researchers have actually tracked this instability down to the molecular level in the Aplysia, right? What does the destabilization physically look like?
It looks like an active demolition site. When researchers reactivated a consolidated memory in the Aplysia by eliciting the gill-withdrawal reflex, they observed a shocking molecular event at the specific synapses storing that memory.
What happened?
The retrieval process triggered the activation of the ubiquitin proteasome system right at the synapse.
Let's define that. What is the ubiquitin proteasome system?
Ubiquitin is a small regulatory protein that acts as a molecular kiss of death. When the cell wants to destroy a protein, it attaches a ubiquitin tag to it. This tag acts as a homing beacon for a massive protein complex called a proteasome.
Proteasome.
Yes. The proteasome is essentially a molecular shredder. It pulls the tagged protein inside its barrel-like structure and rapidly chops it up into tiny, useless peptide fragments.
So when the memory is recalled, the cell actively tags the scaffolding proteins, holding the synapse together, and feeds them into a shredder. The synapse literally begins to dismantle itself.
Exactly. The physical structure of the memory is actively degraded. To prevent the memory from being lost forever, the neuron must immediately activate local translation and synthesize a fresh batch of new proteins to rebuild and restabilize the synaptic architecture.
And what happens if you interrupt that?
If a researcher injects a drug that blocks protein synthesis immediately after the animal recalls the memory, the reconstruction fails. Because the demolition has already occurred, blocking the rebuilding process completely and permanently erases the memory.
So every time I recall a memory of my childhood, I am pulling the structural supports out from under it. And if I get distracted or the context is slightly different, the way it gets rebuilt will be slightly different. The save file is overwritten with subtle errors every single time. That perfectly explains the phenomenon of false memories, or how witness testimonies become corrupted over time. The memory isn't a recording, it's a constant act of active reconstruction.
Exactly. It's a living architecture.
And we've spent a vast amount of time dissecting the Aplysia. We've explored habituation, presynaptic facilitation, the massive cAMP-PKA-CREB nuclear cascade, synaptic capture, functional prions, and the demolition of reconsolidation. It is an incredibly detailed model.
It's the foundation of it all.
But sea slugs are evolutionary ancients. We have to ask, does this exact chemical cascade hold up in something with a more complex nervous system, or is this just a quirk of marine biology? Let's move up the evolutionary ladder to the fruit fly, Drosophila melanogaster.
Moving to the fly is vital because it proves the profound evolutionary conservation of these exact mechanisms. Seymour Benzer and his colleagues were pioneers in this area, demonstrating definitively that fruit flies are capable of classical associative learning.
How exactly do you run a Pavlovian conditioning experiment on a fruit fly? You can't exactly tap its siphon.
True. You utilize their highly sensitive olfactory system. You place the flies in a small training chamber. You flood the chamber with a very specific neutral odor, for example, the smell of octanol.
Okay.
Simultaneously, you apply a mild electric shock to the electrified grid on the floor of the chamber. The flies experience the odor and the pain simultaneously.
Classical threat conditioning.
Precisely. Then, to test their memory, you place them in an apparatus called a T-maze. The maze forces them to make a choice. One arm of the maze is suffused with the odor of octanol. The other arm contains a different control odor they have never been shocked around.
And I assume they avoid the octanol.
Normal wild-type flies will quickly move down the maze and overwhelmingly avoid the arm smelling of octanol. They have learned the association perfectly.
But Benzer didn't just want to prove they could learn. He wanted to find the genes responsible for the learning.
Right. Benzer utilized the power of fly genetics. He bred thousands of mutant strains of flies and ran them all through the T-maze. He was looking for flies that were perfectly healthy, could fly normally, could smell the odor, and could feel the shock, but were completely incapable of forming the memory associating the two.
And when he isolated these learning-deficient mutants, the researchers gave them wonderfully descriptive classic genetics names.
They really did. Dumb, dunce, rutabaga, and amnesiac.
Biologists have the best naming conventions, so what was structurally wrong with the dunce or the rutabaga flies?
This is where the evolutionary conservation becomes undeniable. When geneticists finally map exactly which genes were mutated in these flies, they discovered that every single mutation disrupted a component of the exact same cAMP signaling cascade we just painstakingly mapped out in the sea slug.
You're saying the entire pathway is identical.
Down to the molecular cogs. Let's look at the dunce mutant. To keep the cAMP signal precise, the cell needs an enzyme called phosphodiesterase to rapidly degrade cAMP once the memory signal has passed.
Okay, to clean it up.
Exactly. The dunce mutation breaks this enzyme. Because the cAMP isn't degraded, the background signal is incredibly noisy, and the sharp spike required for learning is lost in the static. The fly cannot learn.
And what about rutabaga?
The rutabaga mutation is even more central. It affects the gene that codes for adenylyl cyclase, the very enzyme that takes ATP and creates cAMP. Furthermore, the rutabaga version of adenylyl cyclase specifically lacks the ability to be superactivated by calcium.
Wait, if adenylyl cyclase is the coincidence detector we talked about in the slug, the enzyme that needs both the serotonin signal and the calcium signal to spike its production, then the rutabaga fly literally lacks the biological hardware to detect the coincidence of the odor and the shock.
Exactly. The fact that breaking the cAMP pathway in a fly completely destroys its ability to form an associative memory proves that this cascade is not a quirk. It is a universal biological law for associative learning.
Let's map the physical geography in the fly. In the slug, the coincidence detection happened right at the synapse between the sensory neuron and the motor neuron. Where does this convergence happen in the complex brain of the fly?
In the insect brain, olfactory learning centers on a prominent bilateral structure called the mushroom bodies. The primary neurons within the mushroom bodies are called Kenyan cells.
Kenyan cells.
Yes. The Kenyan cells are the coincidence detectors. They receive dense, parallel projections from the fly's olfactory lobes, so they receive precise input regarding which specific odor is present in the environment.
That's the conditioned stimulus input. Where does the unconditioned shock input come from?
The shock activates completely separate pathways that eventually stimulate dopaminergic neurons. These dopaminergic neurons cast a wide web, releasing dopamine directly onto the Kenyan cells in the mushroom bodies.
So dopamine takes the place of serotonin in the fly model.
Yes. The molecular detection inside the Kenyan cell mirrors the Aplysia perfectly. The dopamine released by the shock binds to a metabotropic receptor on the Kenyan cell membrane, which, fascinatingly, is the receptor encoded by the DUM gene.
Ah, full circle.
This binding activates a G protein. Simultaneously, the odor signal causes the Kenyan cell to depolarize, allowing calcium to rush in. Both the G protein from the dopamine and the calcium from the odor converge on the adenylyl cyclase enzyme, the rutabaga protein.
And only when both hit the enzyme at the exact same millisecond does it generate the massive learning-inducing spike of cAMP.
Exactly. The mechanics are identical, just the inputs are different. It's plug-and-play biology.
And this brings us to an experiment in the source material that sounds like it was ripped straight out of a science fiction novel. Researchers used something called optogenetic bypassing. Walk me through the setup for this, because it redefines what we consider an experience.
It is a landmark experiment in modern neuroscience. The foundational question was this: Is the painful physical foot shock truly necessary for the fly to learn the association? Or is the shock merely a blunt instrument the environment uses to activate a specific set of dopaminergic neurons?
Right. Can we skip the foot shock entirely?
To answer this, researchers utilize optogenetics, a technique that allows scientists to turn specific, genetically targeted neurons on and off using precise wavelengths of light.
How do you turn a neuron on with light? Neurons run on voltage, not photons.
You have to genetically engineer the flies. The researchers altered the DNA of a specific strain of flies so that they expressed a foreign receptor called P2X. Crucially, they used genetic promoters so that this P2X receptor was only built into the membranes of the specific dopaminergic neurons that project to the mushroom bodies.
Okay, so only the dopamine-releasing neurons have this new receptor. What does P2X do?
P2X is an ion channel normally found in mammals. It is designed to open and cause the neuron to fire whenever it binds to a molecule of ATP floating in the extracellular fluid. But simply expressing the receptor isn't enough because there isn't normally enough free ATP floating around the fly brain to activate it.
So what do?
So the researchers injected the flies with a highly specialized chemical compound, a caged version of ATP.
What does caged mean in a molecular sense?
It means the ATP molecule is chemically bound inside a larger inert molecular structure. While it's in the cage, it cannot bind to the P2X receptor. It just floats harmlessly in the brain.
Okay, I follow.
However, the chemical bonds forming the cage are photosensitive. If you expose the caged molecule to a specific wavelength of laser light, the cage shatters, releasing the active ATP.
Oh, I see the architecture of the trap they built here.
The execution of the experiment is elegant. You take a genetically engineered fly that has the P2X receptors and the caged ATP in its brain. You place it in the training chamber. This fly has never been shocked in its life.
Never felt pain.
Right. You expose the fly to the smell of octanol. And simultaneously, instead of delivering an electric shock to its feet, you simply shine a laser light through the fly's translucent exoskeleton directly into its brain.
The light shatters the molecular cages.
The active ATP is released. It binds to the P2X receptors exclusively on the dopaminergic neurons. The dopaminergic neurons fire violently, dumping massive amounts of dopamine directly onto the Kenyan cells at the exact moment the octanol odor signal is arriving.
Wow.
The coincidence is detected, the cAMP spikes, and the memory is consolidated.
And when you put this fly into the T-maze...
The fly will frantically avoid the arm smelling of octanol. It exhibits perfect, robust threat conditioning.
Wait, so by shining a high-frequency light to chemically mimic the dopamine release, they essentially wrote a file directly to the brain's hard drive without any sensory input from the outside world.
Exactly.
The fly remembers a trauma that physically never happened, that completely redefines what an experience actually is. It's not a ghost in the machine. It's just raw chemical voltage.
It proves definitively that the unconditioned stimulus, the shock, the pain, the physical trauma is nothing more than an evolutionary trigger designed to release a specific neuromodulator at a specific synapse at a specific time.
But if you can replicate the neuromodulator release, the brain cannot tell the difference between reality and the artificial stimulation.
Exactly.
That is profound. It strips the mystique away from memory entirely. We've traced this cascade from marine invertebrates to insects, but human brains are infinitely more complex. How do these exact same molecular cascades operate within the dense, convoluted architecture of the mammalian brain? For threat conditioning, we have to examine the amygdala.
Yes. In mammals, and specifically in human beings, the amygdala is the absolute central hub for detecting, evaluating, and learning about environmental threats.
It's that almond-shaped structure, right?
Yes, an almond-shaped cluster of nuclei located deep within the temporal lobe, nestled near the hippocampus. And its evolutionary programming is incredibly potent. Unlike learning a complex motor skill, which requires thousands of repetitions, the amygdala can permanently associate a new neutral stimulus with a dangerous, life-threatening event after just a single paired exposure.
And it can hold that defensive association for the rest of the animal's life.
It really can.
It's the brain's ultimate survival circuitry. If a rat in a lab hears a specific tone, and that tone is immediately followed by a painful foot shock, the amygdala rapidly makes the connection. How is the internal circuitry of the amygdala laid out to process that association?
Think of the amygdala as having a highly organized input-receiving hub and an output-command hub. The primary input hub is the lateral nucleus of the amygdala.
Okay, the lateral nucleus.
This is where the diverse sensory signals converge. When the rat hears the tone and feels the shock, the auditory information about the tone and the somatosensory information about the pain both travel from the periphery up to the thalamus.
The thalamus is the central sensory relay station of the brain.
Hmm.
From there, how do the signals reach the lateral nucleus? I understand there are two distinct pathways.
Yes, there is a fast route and a slow route. The fast route is a direct projection straight from the thalamus down to the lateral nucleus. This is often called the low road.
And what's the low road like?
It is incredibly rapid, prioritizing speed over accuracy. It provides a rudimentary fuzzy signal that essentially just screams loud noise to the amygdala.
It's the pathway that makes you jump when a car backfires before you even know what the sound is.
Exactly. Then there is the indirect pathway, the high road. This pathway goes from the thalamus up to the highly developed auditory sensory cortex for complex processing, and then down to the lateral amygdala.
Oh, so the high road takes its time.
It's slower, but it provides detailed cognitive context. It tells the amygdala that loud noise is specifically a 2000 Hz continuous tone. Both the fast and slow pathways converge on the dendritic spines of the pyramidal neurons located within the lateral amygdala.
Okay, so the lateral nucleus processes the convergence of the tone and the shock. Where does the output command go?
Once the lateral nucleus associates the inputs, it sends strong excitatory signals to the central nucleus of the amygdala. The central nucleus acts as the master output command center.
And where does it send its commands?
It sends vast descending projections down to the periaqueductal gray in the midbrain, to the hypothalamus, and to various brainstem regions.
And what do those descending signals physically do to the body?
They trigger the full spectrum of physiological defensive states. They command the adrenal glands to release massive amounts of stress hormones like cortisol and adrenaline.
They cause the sympathetic nervous system to spike the heart rate and blood pressure too, right?
Yes. And crucially, they trigger species-specific defensive motor behaviors, which in the case of a rat facing an inescapable threat, is freezing.
Freezing is the evolutionary response to avoid detection by a visual predator. Just lock down and become invisible. So, if the actual associative learning, the memory formation, happens when the tone and the shock converge on the pyramidal neurons in the lateral nucleus, is it using the same long-term potentiation mechanism we touched on earlier with the C-slick?
It relies heavily on long-term potentiation, or LTP. Let's look microscopically at the postsynaptic pyramidal neuron in the lateral amygdala during conditioning.
Okay, zooming in.
When the auditory tone pathway fires, it releases glutamate into the synaptic cleft. The glutamate binds to standard AMPA receptors on the pyramidal neuron. This allows a small amount of sodium to enter, causing a mild depolarization. But on its own, it's a weak signal. It doesn't cause the neuron to fire.
But if the shock pathway fires at the exact same millisecond,
The shock pathway is highly potent. When it fires, it releases massive amounts of glutamate, strongly depolarizing the pyramidal cell. Now we have a coincidence.
the tone pathway is releasing glutamate, and the shock pathway has massively depolarized the cell membrane.
Yes. This simultaneous combination is the specific key required to unlock a completely different type of receptor located at the tone synapse, the NMDA receptor.
The NMDA receptor. This is the famous coincidence detector of the mammalian brain.
Yeah.
How does it work physically?
At resting voltage, the pore of the NMDA receptor is physically plugged by a large magnesium ion. Even if glutamate binds to the receptor, nothing can get through because the magnesium is blocking the door.
So how do you get the magnesium out?
The only way to eject the magnesium plug is by strongly depolarizing the cell membrane.
Which is exactly what the massive shock signal does.
Precisely. The strong depolarization from the shock repels the magnesium plug out of the pore. Because the tone pathway is simultaneously releasing glutamate, which binds to the now unplugged NMDA receptor, the door is forced wide open.
And what goes through the door?
Unlike AMPA receptors, which only let sodium in, the open NMDA receptor allows a massive flood of calcium to enter the postsynaptic cell.
And there's the calcium trigger again.
Yes. Specifically, in the lateral amygdala, this threat conditioning requires NMDA receptors that contain a specific protein subunit called GLO-N2B. Genetic knockout studies in mice have shown that if you mutate or remove the GLO-N2B subunit, the massive calcium influx fails, and the animal becomes completely incapable of learning the threat association.
And once that massive calcium flood enters the postsynaptic neuron, does it trigger the exact same cAMP-PKA-CREB cascade we saw in the slug and the fly?
It triggers an incredibly similar cascade. The calcium activates a series of intracellular protein kinases, including PKA and MAPK. These kinases travel to the nucleus, activate CRE, and initiate the transcription of new proteins.
And what's the final result of all that?
The ultimate structural result of this cascade is the manufacturing of more AMPA receptors. The cell transports these newly minted AMPA receptors to the membrane and physically inserts them into the specific synapse that received the tone signal.
So the synapse physically gains more receiving dishes.
Yes, the synapse is permanently strengthened. The next time the rat hears the tone, the auditory pathway releases glutamate. But now, because there are significantly more AMPA receptors present to catch that glutamate, it causes a massive depolarization all on its own.
So the pyramidal cell fires robustly, activating the central nucleus and triggering the freezing response without the shock ever needing to happen.
Exactly.
Hold on, I understand the mechanics of LTP. The synapse gets stronger. But how do we know definitively that this increased synaptic strength in the lateral amygdala is the memory itself? In complex biological systems, correlation isn't causation. How do we know the LTP isn't just a byproduct like the heat radiating off a car engine rather than the engine itself?
That's the most rigorous, essential question in neuroscience. To prove that synaptic strength is the physical substrate of the memory, we must be able to manipulate that strength directly and observe a corresponding change in the behavior.
Did someone do that?
And once again, optogenetics provided the definitive, elegant proof.
How did they design the experiment?
Researchers genetically engineered rats to express light-sensitive ion channels, specifically in the auditory input pathways connecting to the amygdala. They then trained the rats using a modified threat conditioning protocol.
Using light instead of a tone.
Right. Instead of playing a tone and giving a shock, they delivered a foot shock simultaneously with optogenetic stimulation of the auditory inputs. So, laser light to the brain plus shock to the foot equals learning. The memory is formed. When they shine the light later, the rats exhibited a robust freezing response.
Okay, the baseline association is established. What was the manipulation?
The researchers took those same trained rats and applied a very specific pattern of low-frequency optical stimulation to those synapses. In neurobiology, low-frequency stimulation over a prolonged period induces long-term depression, or LTD.
LTD, what does that do?
LTD is the precise molecular opposite of LTP. It actively removes AMPA receptors from the membrane, weakening the synapse back to its baseline state.
They chemically weaken the connection. And what happened to the behavior?
When they induced LTD at those specific synapses, and then tested the rats again by shining the light, the rats did not freeze. They behaved as if they had never been trained. Inducing LTD effectively erased the behavioral memory.
They formatted the hard drive, they deleted the file, but how do we know the LTD didn't just permanently damage the circuitry?
Because of the final step of the experiment, they took those exact same rats, who now displayed zero memory of the threat, and applied a high-frequency burst of optical stimulation designed specifically to produce potent LTP at those exact same synapses.
Without the foot shock.
No shocks were administered. They simply used the laser to artificially induce LTP, chemically forcing the AMPA receptors back into the membrane.
And when they tested the rats?
The freezing response returned in full force. Inducing LTP successfully reinstated the exact same behavioral memory without any external physical training or sensory input.
Wow.
This proves definitively that the specific strength of the synaptic connection, the sheer number of AMPA receptors at the junction, is the literal physical manifestation of the behavioral memory.
That is profound. You can dial a specific memory up and down with light just by mimicking the mechanics of synaptic plasticity. Now, what about the central nucleus of the amygdala? We mentioned earlier that it's the output command center, but it's not just a dumb relay station that blindly passes the signal along, is it?
Far from it. The central nucleus contains highly complex internal microcircuits that provide essential inhibitory control. Specifically, there is a specialized population of inhibitory cells called PKC delta neurons.
What do they do?
These neurons act as a strict gatekeeper. Under normal resting conditions, these PKC delta neurons constantly inhibit the main output neurons that project down to the brainstem. They keep the system clamped down.
So the default state of the amygdala is brake supplied.
Exactly. In order for the animal to actually exhibit a freezing response, the excitatory signal from the lateral amygdala must do two things simultaneously when it reaches the central nucleus.
What are the two things?
First, it must excite the main output neurons. Second, and crucially, it must heavily inhibit the PKC delta neurons, effectively releasing the brakes and opening the gate.
Why have such a complex system of checks and balances at the output stage?
Because false positives are metabolically expensive and behaviorally dangerous. An animal cannot afford to launch into a full-blown panic freeze every time a shadow moves or a twig snaps. The inhibitory gate ensures that the defensive response is only deployed when the associative signal from the lateral amygdala is overwhelmingly strong and validated.
And how does this highly mechanistic rodent circuitry map to the complexities of human psychology? Because a rat freezing in a plastic box is one thing, but human emotional processing is incredibly messy and heavily influenced by our conscious thoughts.
It maps perfectly, actually, and it brilliantly demonstrates the profound separation of implicit and explicit memory systems that we discussed at the very beginning of the episode.
How so?
To see this in action, we can study rare human patients who have suffered specific, highly localized damage restricted entirely to their amygdala, usually due to a condition called Urbach-Wiethe disease.
So their hippocampus, their explicit memory, is completely fine, but their implicit threat center is destroyed.
Yes. If you set up a standard visual threat conditioning experiment with these patients, for example, they sit in front of a computer screen, a blue square appears, and 2 seconds later, they receive an uncomfortable but safe electric shock to the wrist.
A standard Pavlovian setup.
Right. A neurotypical person will quickly learn the association. After a few pairings, the moment the blue square appears on the screen, their sympathetic nervous system fires up in anticipation. We can measure this objectively by tracking their skin conductance. As they begin to sweat, the electrical conductance of their skin spikes.
And if you ask the neurotypical subject what's going on, they will say, "Every time I see that blue square, you shock me." They possess both the implicit physiological response and the explicit conscious memory of the rule. So what happens when you put the amygdala-damaged patient through the exact same protocol?
The patient with amygdala damage shows absolutely zero physiological response when the blue square appears. Their heart rate remains perfectly steady, their skin conductance is flat, their palms are dry. The implicit, unconscious, associative machinery is completely broken.
And this is the pivotal observation.
If you ask them what is happening, they will explicitly tell you, "I know exactly what the rule is. Every time that blue square comes up, I am about to get a shock."
Wow. They have the conscious factual knowledge of the impending threat, but their body physically cannot react to it because the implicit translation center in the amygdala is offline.
Exactly. And the double dissociation makes it even clearer. If you find a patient with severe hippocampal damage like Clive Waring, but with a fully intact amygdala, their reaction is the mirror opposite.
So what do?
When the blue square appears, they will sweat profusely, their heart rate will spike, and they will show clear signs of physiological distress. But if you stop the experiment and ask them, "Why are you suddenly so nervous?" they will reply, "I have absolutely no idea. I don't know what that blue square means."
They possess the implicit bodily fear, but absolutely no explicit memory of ever being shocked or learning the rule. That dynamic explains an incredible amount of human psychopathology. Think about severe phobias or post-traumatic stress disorder.
Right.
It perfectly explains how a person's body can hold a traumatic memory triggering a massive panic attack at a specific sound or smell, while their conscious mind has absolutely no access to the factual context of why they're reacting.
It highlights the reality that our conscious narrative is only a small fraction of what our brain is constantly processing and reacting to. The implicit circuits operate largely in the dark, dictating our emotional baseline.
Okay, we need to pull back and look at the macro scale. We have spent an immense amount of time deep in the weeds of molecular biology. We've looked at adenyl cyclase, at pKa translocation, at prions, and at the microscopic synapses in the amygdala.
We have.
So, what does this constant synaptic modification mean for the whole brain over the course of a lifetime? If our millions of synapses are constantly growing, capturing proteins, and shrinking based on our daily habits, how does that shape the overall visible physical geography of who we are?
It brings us to the fascinating concept of cortical reorganization. For a large part of the 20th century, neuroscientists believed that the macroscopic maps of the adult brain were essentially static.
Static meaning unchanging.
Right. For instance, the primary somatosensory cortex contains a physical topographical map of the entire body surface, often called the cortical homunculus. We used to think that once this map was wired up during childhood development, the boundaries were rigidly fixed.
But that's not true.
No, the research on implicit learning and skill acquisition proves that these maps remain highly plastic throughout adulthood.
Meaning the physical real estate in the brain, assigned to a body part, can actually change size.
Dramatically so. A classic experiment demonstrated this by studying the brains of adult monkeys. Researchers carefully mapped the exact area of the monkey's somatosensory cortex that corresponded to the sensory input from its fingers.
Okay.
Then, they trained the monkeys to perform a highly repetitive, demanding task. The monkeys had to reach into a tiny rotating cylinder and use only the tips of their middle 3 fingers to repeatedly extract small food pellets.
They forced them to develop a highly specific fine motor implicit habit.
Yes. And the monkeys performed this task for thousands of trials over several weeks. It became a deeply ingrained skill. When the researchers eventually remapped the monkey's cortex, they found a stunning physical transformation.
What happened to the map?
The cortical area devoted to representing those specific middle 3 fingers had expanded massively. The repetitive, intense use of those specific synapses had caused the cortical representation to physically swell, essentially annexing cortical real estate from the adjacent fingers that weren't being utilized in the task.
The brain literally reallocated its processing power to prioritize the most heavily used pathways. It's like a muscle physically growing in response to weightlifting, but on a neurological level.
Exactly. And we see the exact same phenomenon of macro-scale reorganization in humans, particularly in those who dedicate their lives to mastering complex implicit skills.
Give me an example in humans.
The neuroscientist Thomas Elbert conducted brilliant fMRI and magnetoencephalography studies on professional string instrument players, violinists, cellists, and guitarists.
Why string players specifically? What makes them a good model for studying brain maps?
Because playing a string instrument involves a profound asymmetry in motor demands. A violinist uses the fingers of their left hand to perform incredibly complex, rapid, and highly individuated movements on the strings to hit precise notes.
Right.
However, their right hand is used to hold and draw the bow, which is a much simpler, sweeping, gross motor action.
So you have a built-in control group within the same person.
Exactly. When Elbert looked at the somatosensory cortex of these professional musicians, he found that the cortical representation of the fingers of their left hand was massively expanded and highly complex compared to the brains of non-musicians.
And the right hand.
However, the representation of their right hand, the bowing hand, was virtually indistinguishable from a non-musician's.
Because the right hand isn't learning a complex, new implicit habit. It's just maintaining a basic grip.
Right. The thousands of hours of practicing scales and concertos had physically altered the macroscopic topography of their left hemisphere somatosensory cortex.
That's incredible.
And here is an incredibly poignant detail from that study. The extent of this cortical expansion in the left hand was strongly correlated with the age at which the musician began their training. If they started playing before the age of 13, the structural reorganization was significantly more pronounced than if they started later in life.
The plasticity of the brain is far greater during developmental windows, allowing the environment to permanently sculpt the foundational architecture. Which brings us to the ultimate philosophical synthesis of this entire deep dive.
Okay.
We started by asking how experience changes us. Based on everything we've covered, from the slug to the human, what is the biological philosophy here?
The philosophy derived from the cellular biology is one of profound, structurally unique individuality. Think about the implications. Because you, the listener, are exposed to a completely unique set of environmental stimuli. You have a unique set of childhood traumas, a unique set of conditioned threat responses, a unique set of complex motor skills, and a unique set of daily habits. Because of that wildly unique daily experience, your brain has physically altered its macroscopic architecture.
The daily firing of your neurons dictates the shape of the network.
Exactly. Specific synapses have grown stronger through LTP, capturing proteins and building permanent prion structures. Other unused synapses have been pruned away. Specific areas of your sensory and motor cortex have expanded to accommodate the skills you practice, while other areas have contracted.
So your unique biology is shaped by your unique life.
This highly unique physical architecture, combined with your unique baseline genetics, is the literal biological basis of your individuality. No two brains on Earth are structurally identical because no two lives are identical. You are the sum total of your synaptic modifications.
You are the sum total of your synaptic modifications.
Yeah.
I want to pause and synthesize the core takeaways from this monumental journey. We've covered an immense amount of ground, and I want to make sure the foundation is rock solid for anyone listening.
Let's review the key points.
First, we established the fundamental divide. A massive portion of our personality, our emotional baseline, and our daily experience is guided by the implicit memory system, operating entirely outside of our conscious, explicit control.
Yes, the clinical cases of amnesia perfectly illustrate how resilient and isolated the implicit skill and habit circuits truly are. Second, we localized a major hub of that implicit system. In mammals, the amygdala serves as the primary epicenter for both innate and learned threat responses.
And it is a highly efficient machine capable of forming lifelong defensive associations after just a single exposure to a danger.
Right. And the mechanism behind that learning in the amygdala is long-term potentiation. The simultaneous convergence of a neutral stimulus and a strong, depolarizing threat stimulus forces the NMDA receptors open.
The resulting flood of calcium initiates a cascade that physically modifies synaptic strength by inserting new AMPA receptors. The modified synapse is the memory.
And we must emphasize the astonishing evolutionary conservation of that underlying molecular machinery. Whether you are observing a simple reflex in a sea slug, olfactory conditioning in a fruit fly, or complex thmet conditioning in a mammal, nature utilizes the exact same core algorithm.
The cAMP-PKA and CRE cascades are universal biological tools for translating experience into structure.
Exactly. We also saw how human clinical studies validate this circuitry, confirming the absolute anatomical separation of implicit threat conditioning in the amygdala from explicit factual memory in the hippocampus.
You can possess a crippling physiological fear response without any conscious memory of why it exists.
And finally, we scaled those microscopic changes up to the macro level. We saw how habits and skills, when acquired through relentless repetition, fundamentally and visibly alter the large-scale physical geography of our cortical maps, sculpting a brain that is uniquely adapted to its specific environment.
It is a remarkable journey from molecules to personality. Thank you for guiding us through the intricate depths of Chapter 53, unpacking everything from the simplest gill-withdrawal down into the nucleus to watch cREB activate genes, and all the way back out to the philosophy of individuality has been an incredibly enlightening exercise.
It is a profound subject to explore. Understanding the cellular basis of learning fundamentally shifts how you view yourself and everyone you interact with.
It truly does. And I want to leave you, the listener, with one final thought to process. Think about everything we just discussed regarding gene expression, protein synthesis, and synaptic capture. Think about the physical effort required to encode a memory.
It's an active process.
By simply sitting here and listening to this deep dive for the last hour, by actively paying attention, by following the analogies of factories and Lego blocks, and by forming new conscious memories about the intricate biology of neuroscience, as you did that, your brain was physically working.
Constantly working.
Your neurons have literally fired millions of electrical action potentials. You have allowed waves of calcium to rush into your synapses. You have unleashed pKa to travel to your nuclei and activate CRAB1. And right now, in real time, you are physically growing new strengthened synaptic connections. Because of the simple experience of listening to this, you are, quite biologically and literally, not the same physical person you were when you pressed play. You have rewired your own motherboard. Something to mull over. We'll see you.